Hello everyone, Namaskar, welcome to our channel Ashwatha Edu-hub, and today we are going to talk about the history related to photosynthesis. As I had stated in another video, that I'll make a lecture only on the history related to photosynthesis. Okay, so let's see. So when we talk about photosynthesis, it is important for us to know that there has been contribution of a lot of scientists. Today we write the equation what a photosynthesis as carbon dioxide plus water yields what? Glucose, isn't it? And if you balance it, I'm also giving a trick to remember this. So we know the raw materials are what? Carbon dioxide and water, and the products will be what? Glucose, oxygen, and water. So you have to remember the formula of glucose for this. The formula of glucose is what? C6H12O6. So just put a 6 here. C6, H how much? 12O6. Here also it is 6, here also it is 6. So during examination, what might happen? You might take time in balancing the equation. So this is a trick for you to remember the balanced equation of photosynthesis. Now, today we easily write this equation. We know like the evolution of oxygen is what? From water, not from carbon dioxide. But there are many scientists behind this who actually scientifically with experiment they proved certain things. So let us see. So in 1770s, there was a scientist called as Joseph Priestley. Okay, he is the person actually who discovered oxygen with his phlogiston theory. So Priestley he did an experiment with a bell jar with a burning candle and a mouse. So what did he do? He burned a candle, he kept a mouse and covered it with a bell jar, inverted one. Okay. Now what will happen to this burning candle and the mouse? After some time, this will extinguish. The candle will extinguish. It will not continue to burn. And what will be the fate of the mouse? It will die after some time. Clear? After that, what did he do? He introduced a mint plant inside the bell jar. He introduced a small plant inside the bell jar. Clear? Now what happened? Now the candle continued to burn. Now the candle, it continued to burn. It was not extinguished, as well as the mouse was alive. It survived. Clear? Simply by introducing the plant, he found out that the candle was able to burn continuously and the mouse survived. How? He suggested that while burning, the candle made the air impure. Upon burning, the air became what? Impure. What he called it as phlogiston. Clear? Now, when you introduce the plant inside the jar, what happened? The plant, it converted the impure air or phlogiston into what? Pure air or dephlogiston. He knew that somehow plants produce something that makes the air pure. Clear? So this was his phlogiston theory. So from this we come to know that Plants, they produce certain things which can actually help in burning the candle, and it helps the mouse to survive. Now we can easily say what happens actually while the plant was photosynthesizing. It was picking up what? Carbon dioxide, because the burning candle was showing what? Combustion. It was releasing CO2, and that CO2 was picked up by what? The plant, and it was releasing what? Oxygen. And that oxygen actually helped the candle to burn and the mouse to survive. Clear? So there is a question for you. Let me know, what is the origin country of like birth of Joseph Priestley? Do let me know to which country he belongs to. Clear? After this, there was another scientist named Jen Ingenhouse. So he did an experiment in about 1779. Okay, what did he do? He took a bowl with water and he kept hydrilla plant here. Okay, and he kept this bowl under sunlight. Clear? He took similar bowl with water and a hydrilla plant, exactly same replica of this bowl. Okay, and he kept this bowl in dark. Two similar kind of bowl with hydrilla plants in water, one in sunlight, another in dark. Okay, after that what he found, the one which he kept in sunlight, it produced what? It produced bubbles. Here you could see bubbles in water, clear? But in the one which was kept in dark, there was no bubbles. From this, he suggested that what? Sunlight is important for photosynthesis, another important step. This is how actually so many scientists, they have contributed so much towards science. It is not the work of one man, but it is work of many, many, many men and women in science who have contributed what we are living today. Clear? So this suggested what? The importance of sunlight in photosynthesis. Clear? So this Jan Ingenhouse, he was actually a Dutch botanist. Okay. After that, there was another experiment done by another scientist called as Julius von Sech. He made another important discovery. So it was actually Julius von Sech in 1854. He was a German biologist. He suggested that for photosynthesis to occur, there is necessity of what? There is necessity of a specific cell organelle. Now we know that organelle. What is that organelle? That organelle is what? Chloroplast. Clear? And he also suggested what? Suggested that glucose is stored in form of starch. So this is his contribution. Clear? So he suggested that for photosynthesis to occur, there is necessity of a specific cell organelle. Now we know that it's chloroplast, and the product of photosynthesis, which is glucose, it is stored in form of starch. Now let us see another contribution done by another German scientist, T.W. Engelmann. What did he do? He did an experiment with a green algae called Cladophora. He took a green alga Alga is the singular form. If we call it in plural, we call it as algae, G-A-E. He took a green algae, cladophora. Clear? Suppose let us imagine that this is the green algae, cladophora. Clear? And this green algae, cladophora, was kept in a suspension of, it was kept in a suspension of what? Aerobic bacteria. Means surrounding the green algae cladophora, there were a lot of aerobic bacteria. Aerobic bacteria means what? The bacteria which uses oxygen during respiration. Okay. Now, Engelmann, what did he do? He took a prism, okay, and he split light. Now, when light passes through the prism, what do you get? So if you remember, we get what? We get the Vibgyor, isn't it? Vibgyor, the visible spectra of light. Violet, indigo, blue, green, yellow, orange, red. Now what did he observe? Suppose this is the Vibgyor. Okay, he found out that the aerobic bacteria, they accumulated near the blue region and it accumulated near the red region. Why? Because these are the region in which what? Photosynthesis occurs. That is why what happens? The product of photosynthesis is what? Glucose, and with glucose what we find? Oxygen is released. So in those area where oxygen is released or liberated, the aerobic bacteria, they moved towards those wavelengths. So from this he suggested that for photosynthesis, which two wavelengths of light are necessary? The most efficient wavelength are what? Blue and red light. These are the most efficient wavelength for photosynthesis. And he proposed the absorption spectra of photosynthesis. So from his experiment, actually we know that we don't require all the kind of solar radiation, or we don't require all the wavelength of Vibgyor. It is basically the blue light and the red light which is necessary for photosynthesis. So there might be questions on Neet regarding what kind of alga was used by T.W. Engelmann. Answer is what? Cladophora. What wavelength of light, or what are the colors of light which are most efficient for photosynthesis? Answer is what? Blue and red. Clear? Now let's move to another important contribution. There was a scientist called as Van Neel, C.B. Van Neel. C.B. Van Neel in 1931, he made a very important suggestion, and he stated that photosynthesis is essentially an energy dependent reaction. Photosynthesis is essentially an energy dependent reaction in which there is a proton donor. In which there is a proton donor which helps in reduction of carbon dioxide into glucose. So this is a very important thing. Now we know, no? Now it has been established. So C.V. Van Neel in 1931, he is actually a Dutch American scientist. He proposed that photosynthesis is essentially an energy dependent reaction. That energy we know. What is that energy? That energy is what? Sunlight. In case of plants, this is the form of energy. Clear? It's an energy dependent reaction in which there is presence of proton donor. So when water will split, what will be produced? When water will split, it produces what? Proton. This is the proton. Now who is donating this? Water. In case of plants, the proton donor is what? Water. Clear? So, if you look into the actual equation of photosynthesis, Van Neil has contributed so much you get to know from here, isn't it? And he suggested that proton donor will help in reduction of CO2 into carbohydrate. Clear? In the dark reaction of photosynthesis, we will get to know how carbon dioxide is reduced into carbohydrate, that is glucose. Clear? Now, he suggested another important thing, and he suggested that evolution of oxygen is from what? Water. If you look into the equation of photosynthesis, this was the equation. So this is the equation, isn't it? So CO2 also has oxygen, water also has oxygen. So there is like many possibilities who will be releasing oxygen. Maybe carbon dioxide alone is producing oxygen. Maybe water is producing oxygen. Maybe it's combinedly produced by both this and this. But this confusion was actually, experimentally proved later on by to another scientist, Ruben Kamen Hasid. They actually proved it, but it was suggested by C.V. Van Neel that evolution of oxygen is from what? Water, not from CO2. He suggested this. And later on, Ruben, Hasid, and Kamen, they experimentally proved it. So let's see how they experimentally proved it. So in 1941, Ruben, Hasid, and Kamen, they experimentally proved that evolution of oxygen during photosynthesis is from water. So what did they do? They use isotopes of oxygen. Clear? They use isotopes. They took the heavy isotope of oxygen, that is O18. Clear? Now what did they do? In carbon dioxide, they kept the normal oxygen, that is O16, and in water they use the heavy isotope, that is O18. Clear? This is how they experimented. In carbon dioxide, using the normal one, and in water they use the heavier isotope of oxygen. Now, after release of oxygen in the process of photosynthesis, they found that the oxygen that was released, it had heavier isotope of oxygen. So from this they experimentally proved what? It is from water, not from carbon dioxide. So this is actually a brief history you need to actually know while studying photosynthesis. I have already done the part 1 of photosynthesis where I have discussed a bit of the cell organelle involved in photosynthesis, introduction of the mechanism of photosynthesis, and slight part of light reaction. Next video will be uploaded very soon, which will be mechanism of light reaction. See you then. Bye bye.